worked in Sichuan University and did his PhD in Chinese Academy of Science, and then came to the United States in 2005 and did his postdoc in University of Medicine and the Dentistry of New Jersey. He started his independent research lab in Ohio State University in 2012, and his research is currently funded by 4 R01s and 1 AHA award. He has more than 90 peer-reviewed research articles, and his research is pretty broad and covers cancer biology and muscle injury and repair. We both are interested in cardiac damage and repair. Today, he's going to talk to us about how the plasma membrane damage is repaired in myocardial injury. Here's Dr. Zhu.
Thank you, Dr. Li. So thank you, Dr. Li, for your invitation and the kind introduction. So this is my great honor to share one of our project in your university. So the topic of my talk is about cell membrane damage repair in myocardial infarction and beyond. So this is my conflict of interest slides. So what is plasma membrane? Plasma membrane actually is an evolutionary idea. So when the cell is still in the prokaryotic stage, there is no plasma membrane. But when the prokaryotic cells fuse together, the subcellular organelle forms, then the plasma membrane forms. And to protect the cell from the outside insult, and also to keep the cell integrity for the cell energy production and the cell reproduction. However, the plasma membrane is very vulnerable to the mechanical and chemical insults. For example, we know that in the mechanical active tissue like heart, the cell membrane of the cardiomyocyte or some other cell inside of the heart is always face the mechanical injury during the contraction and the relaxation cycle. So if the plasma membrane damage cannot be repaired in time, the cell will die within seconds. Another example is cancer. So think about that. Think about how many cancer patients died from the primary cancer site. It's not many. So it has been established that more than 95% of the patient that die actually is from the cancer metastasized to the remote tissue. So the reason is, at least in my opinion, one of the reasons is because there is a very, very strict selection process for this cancer cell to metastasize to the remote site. For example, a cancer cell will migrate from the primary site and pass through this very tight junction in the blood vessel or in the lymph vessel to travel in the blood circulation or lymph circulation. So this infiltration into the blood vessel will cause a lot of mechanical damage to the plasma membrane of the cancer cell. And the inside of the circulation, the cancer cell will bump into different blood cells, like white blood cell, red blood cell, and those blood cells are designed for traveling through the circulation, but not for the cancer cell. So then finally the cancer cell will squeeze out from the blood vessel or lymph vessel to form the distal metastatic cancer. So in this process, actually, it's highly, highly inefficient. The reason for inefficient, I already mentioned that, is too many stress for this metastatic cancer cells. So it has been established that more than 99% of the cell that migrate and metastasis remote tissue will eventually die. So that only about 0.1% of cell will survive during this highly stressful condition. So in here, I think plasma membrane damage repair plays another huge role here. So what's the mechanism for the plasma membrane damage repair? I borrowed this movie from Dr. Paul McNeil, who is a pioneer in this field. So you can see from this movie, and there are a lot of intracellular vesicles. By the way, he used the sea urchin egg as a cell model for this experiment. So you can see that inside of the cell, there are a lot of intracellular vesicles. And those intracellular vesicles serve as a membrane reservoir to patch the membrane. Whenever the cell has the plasma membrane damage, the calcium will get into the cell, and the intracellular vesicles will travel to the damaged site, and because of the calcium, they will fuse together to serve as a membrane patch to patch this hole. After that, the membrane patch will finally fuse together with damaged plasma membrane to finish up this membrane damage repair process. So our study is focused on a very unique structure, so which is very important for the excitation contraction coupling in skeletal muscle. So this unique structure is called triad structure. So more than 20 years ago, our long-term collaborator, Dr. Hiroshi Takashima in Kyoto University, developed this type of experiment to identify the triad structure protein in the rabbit muscle. So he injected the triad structure protein into mouse, from the rabbit into the mouse, and to generate the monoclonal antibody to identify the antigen of those antibody. Then he cloned different newly identified genes and generate the mutant mice to study those proteins function in the excitation contraction coupling. After his pioneer study, he identified a lot of novel protein resident in the triad structure. One of the protein called MG53, misogumine MG53. So when he found this protein, and he had a very good antibody, he generated the knockout mice. Unfortunately, the knockout mice seems very normal in terms of the normal physiology. So at that time, we don't know exactly what happened in there. So then one day we perform an experiment. We just overexpress this protein, tag this protein with GFP, and perform a live cell imaging. And then we found that a lot of this protein are resident in the intracellular vesicles, and they are traveling around the inside of the cell with the live cell imaging. So because we attend Dr. Paul McNeil's presentation, and his presentation showed that those intracellular vesicles might be contributing to the plasma membrane damage. So the next experiment we did was to introduce the plasma membrane damage with a patch clamp electrode, just to poke the plasma membrane and track the GFP labeled MG53 upon the plasma membrane damage. So here is what we found. When we poke the plasma membrane, we can see the GFP labeled MG53 is traveled to the damaged site within seconds. And if this is a myofiber, if we just cut this fiber into half, we can see that GFP MG53 travel to the damaged site again. And this is another myofiber. We poke a hole here, and we can see the GFP labeled MG53 move to there. So what is MG53? MG53 actually is a trim family protein, tripartite motif family protein. It has the human MG53 protein has the force 77 amino acid. It has a ring domain, B-box domain, and the coil domain. Those three domains form the tripartite motif. And in addition to that, MG53 also have a spray domain here. So, also we show that MG53 can translocate to the plasma membrane damage site, but it doesn't mean that the MG53 will repair the membrane. So how we can prove that? Here is what we do. So here is what we do. We just use the same electro needle to poke a hole on the Y-type muscle fiber. You can see that there is nothing happen. And we move this needle to another site. We poke a hole, and there is nothing happen. And we poke a hole again in the same position, and withdraw the needle. There is nothing happen. So people always take for granted that this will not happen because the biophysical property of the plasma membrane will automatically seal the damage rapidly after the withdraw the needle. This is well observed in the field of electrophysiology because people use the patch clamp to patch the cell all the time. However, this is what we observe in the cell from MG53 knockout mice. So we poke a lot of cells. Almost every cells from the MG53 knockout mice show this property. This experiment indicates that MG53 does play a role for plasma membrane damage repair. If without MG53, the membrane cannot be repaired and the cell will die right away. And after a series of study, this is what we currently understand for MG53 mediated plasma membrane damage repair. So in addition to the calcium observed from the Dr. Paul McNeil's pilot study, we feel that there are two main factors that is involved in MG53 mediated plasma membrane damage repair. One is the oxidative status. Outside of the cell, usually it's oxidized status. Inside of the cell, usually it's more reduced environment. Another important parameter is the lipid. It's called phosphatidylserine. So phosphatidylserine is usually embedded in the double lipid layer membrane. Only upon the damage, phosphatidylserine will expose to the damage site of the plasma membrane. We believe that serve as a plasma membrane damage marker for the recognition of MG53 because MG53 has a high affinity to PS. In addition to that, at the damage site, the oxidized condition penetrate into the cell, it trigger a disulfide bond formation in between of two individual MG53 protein. They serve like hand in hand to pull the intracellular vesicles tightly to the plasma membrane. Finally, as mentioned by Dr. Paul McNeil's pilot study, the rush in of the calcium will trigger the fusion of the plasma membrane, and this fusion event forms the patch of the membrane repair patch to perform the final repair process. As we know that there are a lot of diseases is caused by plasma membrane damage repair, including myocardial infarction. However, if you would like to apply MG53 into those applications, you cannot actually overexpress MG53, just like what we did in the previous slides, to overexpress that, then to protect the cardiomyocyte from the plasma membrane damage during myocardial infarction, because it's too slow. This overexpression system is too slow. So at that time, our thinking is whether we could purify recombinant MG53 protein and apply this in the through the IV or through some other routes to deliver into the body to make this repair happening. So this is what we did. We isolate the recombinant MG53, and we develop ELISA assay to just verify the activity, the plasma membrane damage repair activity of the protein, and we perform the experiment. So here are two individual skeletal muscle fiber, and one fiber we incubate recombinant human MG53 at the culture medium. Another fiber we just use the BSA as a control, and then we introduce the plasma membrane damage with laser beam. So we just shine a laser on the plasma membrane of the skeletal muscle, and we incubate those skeletal muscle with a dye which can stain the membrane damage site. So when the membrane damage happens, the dye will light up, and we can quantify the dye of the membrane. Here is what we found. So in this case, you can see after the laser beam shining, we can see a lot of plasma membrane damage happen because a lot of dye light up. Also, the damaged muscle fiber become curved, bent. And this is what we observe when we incubate the muscle fiber with MG53 at the culture medium, and we see much reduced level of the plasma membrane damage. That's indicate that application of MG53 outside of the cell will also serve as a protective factor for the membrane damage. So the next we test the experiment with the Langendorf system. So here we just hand the heart in the Langendorf system and stop the perfusion to mimic ischemic condition, and to restore the perfusion to mimic the perfusion injury condition. And then we apply MG53 in the perfusion solution to just deliver MG53 directly to the heart. As you can see that the infarct area quantification show that when we add MG53 either before the ischemia or after the reperfusion, both way we can greatly reduce the infarct area of the Langendorf hanging heart. Also, we can connect the perfusate to quantify the creatine kinase level in the perfusate. We can see that either pre-incubation of MG53 or post-incubation of MG53, both way can greatly downregulate the release of the creatine kinase. So next, we perform the in vivo ischemic reperfusion injury in the rat heart. This time, we deliver either rhMG53 or BSA as a control through the IV injection with different dose. And you can see that with different dose, we can see the dose-dependent reduction of MG53 in terms of controlling the infarct area in the rat heart. And also, we perform the experiment in the pig heart. In this case, we introduce the IR injury with angioplastic balloon, and you can see that we can inflate. So we can inflate the balloon in the LAD of the porcine heart to introduce the ischemic condition for a while, and then we deflate the balloon and introduce the reperfusion injury to the porcine heart, and we deliver the MG53 protein either before we do the angioplastic balloon or after we withdraw the balloon to introduce the reperfusion injury, or even we introduce MG53 30 minutes after withdraw the angioplastic balloon. And you can see that from the infarct quantification, we can see that all of those three treatments can greatly reduce the infarct area in the heart as compared to the saline control. And this is the serum quantification of troponin I release in response to the IR injury in the porcine model, and you can see that all of those three conditions is greatly reduce the troponin level in the circulating after the IR injury. We also perform the histochemical staining of the tunnel staining to quantify the apoptotic cell after the acute injury at different group. And you can see that treatment of MG53 greatly reduce the tunnel positive cells in the porcine heart. Interestingly, when we take out the heart 24 hours after injury, then we separate the heart with the infarct area, adjacent area, and remote healthy area. And you can see that even 24 hours after we take out the heart, we will see the MG53 treated heart has elevated MG53 level specifically in the infarct area, which is really, really interesting for us because consider the half-life of the recombinant human MG53 in the blood serum, in the blood after the IV injection is only about 2 hours. So we also performed the molecular signaling study to show that after injection of MG53, actually MG53 can activate the AKT GSK3 beta pathway to serve as a pro-survival, to just activate the pro-survival pathway to prevent the cell from death after the myocardial infarction. However, MG53 don't regulate the phosphorylation of ERK. So we conclude that treatment of MG53 is activating the risk signaling pathway for the cardiac protection. We also, after the surgery, we also just maintains the peak for 4 weeks. 4 weeks after surgery, we sacrifice the porcine, and then we dissect the heart out, and you can see that in the saline treatment group, the heart is very dilated, and the cardiac wall is very thin. So indicate there are a lot of myocardial infarction happen, and there are a lot of cardiomyocyte deaths after IR injury. However, when we open up the heart of the group from the pre-treatment of MG53 and the post-treatment of MG53, only from the morphology you will see that the heart wall is thick, and there are still a lot of myocardium left after the injury for 4 weeks. We also detect the fibrotic remodeling in the infarct area, in the infarct area of the heart, and you can see that in the saline group there are a lot of fibrotic remodeling, which is correlated quite well with the cardiac pumping function. And with treatment of MG53, there is greatly reduced fibrotic remodeling in the MG53. We do have a follow-up study to study the molecular mechanism, how MG53 controls the post-fibrotic remodeling after injury. So as a small summary, we can see that MG53 can really protect the plasma membrane from against the damage after the ischemic reperfusion injury. However, it is well known that myocardial infarction induces the reactive oxygen or oxygen species, which will induce the oxidative stress, and this oxidative stress actually the primary target for those oxidative stress is a subcellular organelle called mitochondria. So our next question is whether or not MG53 play a role in this process. If it play a role, then what's the mechanism for that? So even before this, our finding, so Dr. Tish Murphy from NIH already published a paper in the PNAS, and she found that very interestingly, MG53 translocate to the mitochondrial fraction after IR injury. And she labeled the MG53 as TRIM72. As I mentioned to you, MG53 is tripartite motif protein, and the gene name for MG53 is TRIM72. So it's the same as MG53. So she already observed that after the injury, after the myocardial infarction, after the IR injury, the MG53 already can translocate to the mitochondrial fraction. Very similarly, our own study also indicates this similar effect. However, in our process, this is a process of the high-fat diet induced oxidative stress. So at that time, a high school student in my lab was performing his summer study in my lab, and after his study, he basically first feed the mice with high-fat diet. Then he just sacrificed the mice and stain the skeletal muscle of the mice from the normal diet or from the high-fat diet. Then he came to me and told me that, Dr. Zhu, I observe a little bit different subcellular distribution of MG53. As you can see here, the MG53 is evenly distributed throughout the skeletal muscle fiber. And in this condition, MG53 is form a small circle in the skeletal muscle fiber fitted with high-fat diet. And he told me that every fiber, those small circle, inside of those small circle, there is DAPI positive staining. So at that time, we already thought about this because everybody knows that the nucleus of the skeletal muscle is always in the peripheral position. So nucleus of the skeletal muscle cannot be in the central of the muscle, otherwise the muscle will have some disease. So therefore, this type of DAPI positive staining could be another possibility, which is mitochondria, because mitochondria also have its independent mitochondrial DNA, so which can be stained by the DAPI. So what I asked him to do is just stain the mitochondrial marker. This time we use COX-4. And you can see that almost every circle of MG53 positive circle is surrounding the COX-4 positive signal. When we blow out this picture, we can see that indeed the green signal is surrounded by the red signal, which is MG53. That is indicate that high-fat diet induced oxidative stress might cause translocation of MG53 to the membrane or to the outside of mitochondria. Similarly, we have another animal model. We collaborate with Professor Jinsong Zhou in UT Arlington, and from her lab, she has a mouse line which can mimic ALS disease. So it is well known that ALL is a disease caused by mutation of SOD1 that causes the huge oxidative stress in the neuromuscular junction, which causes the motor neuron problem and causes the muscle atrophy and muscle dysfunction. And this is the muscle from the ALS mice. We observe the similar effect of the translocation of MG53 in the surrounding the mitochondria. And then let's go to check, because we have our pulsar model of the heart, we have the tissue slides. Then we just go ahead to stain our tissue slides from our pulsar model. And also we can see that this in the infarct area, MG53 staining is colocalized quite well with COX-4 staining, indicate that after IR injury in the heart, MG53 also, part of the MG53 also target to the mitochondrial fraction. So then our question will be, since we introduce the MG53 outside of the cell, we introduce MG53 through the IV injection. So our question is whether MG53 could, how MG53 could target mitochondria if it's introduced outside of the cell, and what's the mechanism for this protection against the mitochondrial injury. And this is a study we perform the live cell imaging. In this case, we incubate the cells with the Alexa 647 dye labeled recombinant MG53. We apply MG53 outside of the cell, and we observe whether those MG53 can penetrate into the cell after this, after the incubation. And this is, oh, sorry. This is real-time imaging, real-time movie. And you can see that there is a very active uptake of MG53 into the cell. That indicates that indeed MG53 can be uptake into the cell. Then we perform the hypoxia reoxygenation injury to the freshly isolated cardiomyocyte, and you can see two observations. First observation is, since we use the enzyme to digest the cardiomyocyte from the mice, and you can see that a majority of MG53 is located on the gap junction side of the cell. The gap junction side is the I believe it's the most damaged side, because gap junction has to be enzyme digested in order for us to get this single cardiomyocyte. Another interesting observation we found is the MG53 actually can be uptake into the intracellular portion of the single cardiomyocyte after the hypoxia reoxygenation injury. And this localization of MG53 is co-localized with the vital tracker deep red staining quite well, indicate that at least a portion of MG53 is located to the mitochondria upon HR injury. So then what's the mechanism? As I mentioned to you, for the plasma membrane damage repair, there are at least two very distinct factors that mediated the MG53's targeting to the damaged membrane. First is the lipid, which is phosphatidylserine, serve as a damage molecule that can target mitochondria. And the second is the oxidative stress that mediated the MG53's dimerization and to target the membrane, damaged membrane. We don't think there is any problem for the oxidative stress induced the oxidative status, which can mediate the MG53's dimerization, because the mitochondria is a primary subcellular organelle that is a victim of IR injury-induced oxidative stress. For that, we don't think that's a problem. However, there is a problem for the lipid, because the membrane of the mitochondria, the composition of the mitochondrial membrane is so different from the membrane of plasma membrane. So from the literature search, we can clearly see that there is no phosphatidylserine present on the membrane of mitochondria. However, with the literature search and the discussion with the expert of the mitochondrial research, we know that there is another lipid. It's called cardiolipin. Cardiolipin is usually present in the inner membrane of the mitochondria, and only after the oxidative stress, the cardiolipin will be oxidized and being flipped from the inner membrane to the outer. The oxidized cardiolipin will be flipped from the inner membrane to the outer membrane, kind of serve as a damage signal for the damaged mitochondria, similar to the role of phosphatidylserine. So our question is whether MG53 can also recognize cardiolipin. So this is what we did. So this is a lipid overlay assay we did. We purchased this lipid strip from the Lisanti company, and we incubate the lipid with recombinant MG53 protein and to perform the Wilson blot. And you can see, indeed, MG53 can recognize very strongly together with phosphatidylserine. And interestingly, MG53 can also have binding to the CL. CL means cardiolipin. So I need to indicate that this cardiolipin is not oxidized form. So this is regular cardiolipin, and we ask the company, and the company said they cannot apply us for the oxidized cardiolipin. So we don't have the oxidized cardiolipin binding assay yet, but we probably will try to do that by ourselves. And we also perform the ELISA assay, and we can see that the binding affinity of MG53 with cardiolipin is kind of high. We compare this binding affinity with the PS binding affinity. We find that the cardiolipin binding affinity to MG53 is even a little bit higher than the phosphatidylserine. So here we all show that MG53 is translocated to mitochondria in different pathophysiological conditions, including myocardial infarction, including ALS, including high-fat-induced metabolic disorder. However, again, it doesn't mean that MG53 protect mitochondria. So how we determine whether treatment of MG53 can protect mitochondrial damage after the injury. So here we use a system which can measure the generation of the RAS generation specifically in mitochondria. And this system gave us a very useful system for us to do this experiment. So basically, myself and another member of my lab, we personally traveled to the lab of Dr. Jinsong Zhou's lab to perform this study by ourselves, because Dr. Zhou has the transgenic mice which overexpress this fluorescent dye we call the flash. So basically, this flash dye will light up whenever there is mitochondrial RAS production. So by quantifying the light up of this dye, we can quantify how much of the RAS produced in response to different injury conditions. So here we introduce the injury with hypoxia reoxygenation injury, and then we either incubate the cell with control or incubate the cell with MG53. And you can see from the control cell, after we introduce IR injury, you can see a lot of GFP positive signal light up. That's an indication of the RAS generation in response to hypoxia reoxygenation injury. We perform the same hypoxia reoxygenation injury and incubate the cell with RhMG53. This is what we observe. As you can see that we definitely observe some part of the flash happening, like here, or some part of the flash happening. However, the frequency and the amplitude of the flash is much reduced after MG53 treatment. This is a quantification of our movie, and you can see the amplitude is high. The incidence of those flash events is high, and after MG53 treatment, the flash gets reduced drastically. So in summary, that we find that MG53 can actually protect the heart against ischemia reperfusion injury, and MG53 preserves the cardiac function after IR injury. And another possibility is MG53 can translocate to the mitochondrial fraction to suppress the oxidative stress by targeting cardiolipin in the damaged mitochondria. And the incubation of the MG53 protein can suppress this mitochondria-generated RAS production to serve as a protective factor for this process. And we recently also have different experiment to show that even in addition to that, MG53 can serve as a protective factor to inhibit the cell death from the cell by suppressing necroptosis, and maybe by suppressing pyroptosis to protect the cardiomyocyte from the death. And if I have chance, I can share with you those stories in the future. So with that, I would first thank you for your attention, and the surgery and the cell work was done by Tiho Part and Bin Chuan in my lab, and the mitochondrial stress part is done by very talented high school student Henning Ma in my lab. Now he's already in medical school now. And I would also like to thank my previous mentor, Dr. Genji Ma and Dr. Hiroshi Takashima. And both of them contribute a lot to the discovery of the MG53's role in the plasma membrane damage repair. I would also like to thank my collaborators for the FLASH transgenic mice LOS study and for the porcine surgery model. And also I would like to thank my funding sources, and thank you for your attention, and I would like to take questions.
Thank you, Hua. This is a very interesting talk, and the data is very fascinating. So I think now it's open for questions. I maybe ask a first question. I'm wondering, we all know the precondition can protect the ischemic damage. Is the MG53 upregulated during preconditioning?
Yes, it has been published by 2 papers from Peking University. Yeah, so they found out that the cardiac preconditioning will upregulate the level of MG53, which might contribute to the cardiac protection function of the preconditioning.
Have you done a timing post MI or post ischemic? What's the the early part is like reopening the coronary arteries. The earlier is better, but have you studied a time? What's the best time to give the MG53? Also, what time it won't be affected anymore?
That's a good question. So the latest time we apply MG53 is after we draw the angioplastic balloon for 30 minutes. So we found that the 30 minutes still have effect, but the porcine model is very expensive, so we didn't test longer time. However, we collaborate with GSK, the company, and they perform the study with ischemic skeletal muscle injury, so the ischemic limb injury, and they found out that they perform a post-injury injection because they have more resource, and they found out that only so the application of MG53 will be inefficient 8 hours after the ischemic limb injury. So they perform the they inject MG53 after the limb injury for 1 hour, 2 hour, 4 hour, 6 hour, and 8 hour. And they found out that only the MG53 will become inefficient when the MG53 is introduced to the skeletal muscle 8 hours after injury. So before that, it will still have protective effect. That's their data.
Thank you. Anyone can have a question, can unmute yourself and then ask the questions.
This is Dr. Dayton. I was waiting for Dr. Furcht to ask this question, but I'll ask it, and which is anyone working on preparing for phase 1 human trials?
We are, one company is called Train Medicine. So they are preparing the phase 1 clinical trial with the application of MG53 now, yeah, in our hospital. Yeah.
Thank you.
Thank you.
Other questions?
This is Khalil Ahmed. I was wondering, what are the calcium dynamics in your system with MG53?
That's a really, really good question. So let me show you the structure of the protein. So you can see that MG53 is a typical trin family protein, and after the trin motif, there is a spray motif. If everybody is very familiar with the spray motif, this motif is presented in a lot of calcium proteins, including RYR and some other proteins. And we do have a couple studies to show that after incubation of MG53, there are a couple different channels, including RYR, being activated by the treatment of MG53. And this activation might be because of their direct interaction.
Any other questions? I may ask a last question. I think that in addition to plasma membrane damage, I think during the mechanic stress or others, and the nuclear membranes are also damaged. Does this protein target to nuclear membrane?
Yeah, one of my we don't have the solid data yet, but one of my member in my lab, he is actively testing this effect. Yeah, so this is also really critical for cell protection.
Okay, thank you. If anyone has questions, you can email me or directly email to Dr. Zhu. He can answer you. I think this is kind of very interesting. It's relevant to human disease, and I hope this can really protect the ischemic damage and in that way save a lot of lives. Thank you all for attending.